In this video, we're going to talk about Mendel's law of independent assortment, which he was able to derive based on his experiments with pea plants, garden peas. Okay, so before we go into this law of independent assortment, I want to remind you about the first law that we learned, which was the law of segregation, and he was able to derive those using the experiments looking at only one gene. So he looked at the gene that determined the flower color, either purple or white, and then the question he asked was that, okay, if I follow one gene and there's variations of that gene, so in this case we have purple and white, there's two different versions of the gene, how are these two versions inherited? And he did the experiments and he was able to derive the law of segregation, and he came out with these ratios. Now, what is law of independent assortment refer to? Well, after he did all these experiments, then just like any good scientist, he wanted to ask the next question. And the question was, well, what if I look at the inheritance of two different genes? What if I look at two different characters? Is the inheritance of one dependent on the inheritance of others, of the other genes, or are they independent of one another? Another way to think about this, if you like, is that if you ask, will all the people that have blue eyes also have blonde hair? So if you have blue eyes and blonde hair and you have children, are all your children going to have blue eyes and blonde hair? And from our life experience, we really know that really the answer to this, it depends. It depends on what kind of genes you inherited, what did your parents or grandparents look like, and also it depends on who you're going to marry, because you could have blonde eyes and blue hair, but you could there is very likely, depending on who you marry, that you're going to have kids that have brown hair and blue eyes, or brown eyes with blonde hair. So we know this right now, but at that time, those questions haven't been answered. So that's what Mendel wanted to figure out. So how did he do it? He looked at two different characteristics in these garden peas that he had. And this time he looked at the shape and color of seeds. So there are two genes. One determines the shape. It's either round or wrinkled. And the other gene determines the color. This seed is either yellow or green. And just like his previous experiments, he set up his experiment with the parental generation, true breeding parental generation. So this parent would only give rise to round and yellow seeds, and this parent would only give rise to wrinkled and green seeds. And then he asked, okay, what happens if I cross these two together? And when he got the F1 progeny, all of them had yellow round seeds. But this is something he would expect anyway, because he knew which version of each gene is dominant over the other. Yellow is dominant over green, and round is dominant over wrinkled. Then the next question he asked was, what if I now cross the F1 with itself? What happens in the next generation? Will yellow seeds always be round? Will green seeds always be wrinkled? What happens? So here's the cross. So we have the P generation, the parental generation, which is homozygous for both genes. They're true breeding, yellow round and green wrinkled. And the F1 generation is a heterozygote. Now, if you cross this heterozygote with each other, what will you get? The first hypothesis would be based on the assumption that yellow and brown seeds would always move to the same gamete during meiosis. So yellow always sticks with round. The next hypothesis was that yellow and round can either move to the same or to different gametes during meiosis. So if we had the hypothesis 1 correct, if always yellow stuck with round, then you would not expect to see yellow wrinkled seeds, or you would not expect to see green round seeds. In this case, you would be able to find those combinations. And when he did the experiment, he was able to see that you could get all the combinations of these two genes. He was able to get yellow round seeds, yellow wrinkled seeds, green round seeds, and green wrinkled seeds. Now, just like that monohybrid cross, in this case, no matter how many times he did the experiment and how many different characteristics he looked at, he always get the same ratio of different variations. We call this kind of cross where you're looking at two different genes at the same time, inheritance of two genes at the same time. We call it the dihybrid cross. So this is a classical Mendelian dihybrid cross. F1 always is 100% showing the phenotype of the dominant versions of the genes you're looking at. But in the F2, you get all of them, but always in a particular ratio. And the ratio is always, let me see if I can write it, is always 9 to 3 to 3 to 1. So 9, this looks like a 4. So, and this is 9 out of 16. So it's a ratio, it's not an absolute number. So all of these are out of 16. Okay. So the ratio is 9 to 3 to 3 to 1. 9 out of 16 are going to have the phenotype of the dominant allele for the two genes. 3 out of 16 will have dominant for one and recessive for the other. Yellow is dominant, but wrinkled is recessive. The other 3 out of 16s have the alternate situation where you have, again, a dominant for one gene and recessive for another one. And 1 out of 16 has both of them recessive. So what does that look like? How do you get a ratio like that? If you do the cross, you will see. So we have this F1 generation, and we're going to cross this to another F1. So it's as if you have two of these crossing with one another. So you would have a big Y, little y, and big R, little r. So you have two of them. And each of these parents are going to get 4 different gametes. Each of these are going to make 4 different gametes. And you put it on a Punnett square and you score the phenotypes, and you will see that you get a 9 to 3 to 3 to 1 ratio. For a dihybrid cross, this is a dihybrid cross. In a classical Mendelian dihybrid cross, the ratio you would expect in the F2 is always 9 to 3 to 3 to 1. For F1 is always 100% of the dominant genotypes. Dominant phenotype. So, now here's a step where you get to practice. So what does independent assortment mean? So what is the law of independent assortment and how is it related? Basically, independent assortment means that when the time comes for meiosis to produce egg or sperm, the way that homologous chromosomes sit in the metaphase plate is independent of one another. That means you can have this combination of all blue on one side and all red on the other side. But then this pair of chromosomes can flip. And it's totally independent of how this pair sits in the metaphase plate. And this rises to a situation where you get all 4 different combinations. So draw these steps with the gene of seed shape on the large chromosome and the gene for seed color on the small chromosome. And then you will see the types of combinations of gametes that you will get. Okay, so next thing that we're going to learn is a test cross. So what is a test cross? It's a tool for geneticists to find out the genotype of an organism that presents a dominant phenotype. So we know that for a dominant phenotype to present itself, the individual could be either homozygous dominant. So in the case of this yellow, we could have big Y, and then you would have yellow seed. I don't really write well with this thing, but I'm doing my best. Or you could have a big Y, little y. That's a definition of a dominant allele. You only need one allele for it to be expressed. So again, this is going to be yellow, just like above. So this symbol means that I'm copying from here. It's exactly the same thing. So if you get an individual that is expressing a dominant phenotype, you can't be really sure of its genotype, because it could be this one, or it could be that one. So how do you solve that? The way scientists do it is they cross the unknown individual with a homozygous recessive. And then look what happens. So if the unknown is homozygous dominant and you put the possible gametes from this individual on a Punnett square and from this one, you will see all the progeny are going to have a yellow seed. That means all the progeny are going to show the dominant phenotype. So 100% of the phenotype of the progeny would be the dominant trait. Dominant. It's not easy to write with this. Dominant. Okay? So if the unknown is homozygous dominant, all the progeny of this test cross will have the dominant phenotype. But if the individual is heterozygous, okay, then, and you do the Punnett square, you see 50% will have the dominant phenotype and 50% will have the recessive phenotype. So you have a 1-to-1 ratio, or 50% to 50%. 50% will be dominant phenotype, 50% will have a recessive phenotype. So that's a test cross. It's an easy way to do that, especially with organisms where you can set up crosses at will. So if you have an individual who's expressing a dominant phenotype, you don't know their genotype, you cross them with a homozygous recessive. If the individual is the genotype of the individual, which is unknown, has a homozygous dominant genotype, then all the progeny are going to show the dominant phenotype. If the unknown is heterozygous, then you get a 50-50 of recessive to dominant ratio.